At Endless, we strive to stay ahead of the curve in both cannabis science and disease testing, and TUMIGenomics shares the exact same mindset. Today, we had the opportunity to sit down with Tassa from TUMI and discuss how they've been able to support us in the achievement of this goal and their very unique journey, and how it began with cancer research and evolved into cannabis disease testing. We hope you enjoy.
Endless strives in being ahead of the game, and disease testing is one of those things that we try to catch pretty early on. And with TUMI's amazing products, they help us do just that. We're happy to be sitting down with Tassa from TUMI and Madison from Endless to discuss some disease testing and how Endless and TUMI kind of coexist together. Madison, thanks for joining us on this journey and this call.
Absolutely. Thanks for having me. I'm excited.
Yeah, Tassa, thank you for joining us as well.
Yeah, no, absolutely. I'm also excited. I think this is going to be a great conversation.
For sure.
Cool. So let's get it started with Tassa. If you want to kind of introduce yourself, what you do for TUMI, and kind of some of the things you see on your day-to-day basis.
Yeah, absolutely. So I'm Tassa. I am one of the co-founders of TUMIGenomics, as well as the chief science officer of the company. And so TUMIGenomics, we founded the company about 4 years ago, and or 5 years ago now. And our main goal is to help cultivators mitigate pathogenic stress in their facilities. And so we provide, obviously testing and pathogen diagnostics, as well as a lot of remediation and mitigation guidance. And so we kind of pride ourselves on working with cultivators and following them through their entire journey. From the beginning of, either setting up a preventative program or realizing they have a problem, all the way through figuring out how to solve that problem, how to change what's going on in their grow, and sort of leading them to success.
Absolutely love that you mentioned you're part of their journey, and that's one thing that's huge, making sure that you stay with those customers and you understand what it is they're doing. I know with Madison, you talk a lot about the communication between you and TUMI, and how you speak so highly of their team. So with that, let me ask you, Madison, what is it that you do on a day-to-day basis? I know you do so many different things. Can you just sum it up into a few short sentences?
Yeah, I do wear a lot of hats, but so right now my primary function is kind of a hybrid role between cultivation manager and overseeing operations in our laboratory. But one of the things that has always been a key aspect of my role at Endless is disease testing, and it remains true to this day. So having the TUMIGlow having TUMIGenomics as a resource has been extremely valuable for pathogen detection, early detection, all that good stuff. But yeah, as far as my role goes, a little bit of everything, but disease detection and prevention is even if it seems like a small fraction of my day-to-day, it's a huge part of making sure that our cultivation operation is successful. So yeah,
absolutely, many hats, many hats,
all the hats.
So Tassa, I want to ask you a question. So what kind of inspired you to begin TUMIGenomics, and was cannabis the main focus whenever you started this company?
I would say, as far as the inspiration, we were founded by a group of postdocs and some friends that had some entrepreneurial experience. And basically, we were kind of done with academia, and we decided that we really wanted to take what we knew how to do and apply it to, an industry or a geography that really needed, high quality science and tools, to help them in some capacity. And so originally, cannabis wasn't, where we started originally. We actually started in cancer diagnostics, of all places.
Wow.
Oh, wow.
And so we wanted to do molecular cancer diagnostics. But very quickly, COVID kind of hit and the world changed, and we pivoted into doing COVID diagnostics. And kind of while we were in the thick of that, we had one of the people who founded the company had a friend who runs a nursery in California, and she was having issues, diagnosing this crazy new pathogen that had infected her grow and finding accurate testing, and was like, hey, maybe they could help me, on the side. And we were like, sure, it's an RNA, we do RNA biology, that's fine. And it was like all of these light bulbs went off. We designed the test to help them out, and then she told a few other people, and it just kind of exploded from there. And I would say in some ways the cannabis industry kind of pulled us in.
Yeah.
Yeah. But once we found it, it was the perfect place for us to be. I mean, if there ever was an industry that needed more high quality science, more research, and education, and kind of a trusted information based on published research, I think cannabis was it. And so it ended up being an awesome industry. And I love working in the cannabis industry. People are very cool.
Awesome. One of the words you mentioned a second ago, and it might be brought up multiple times, is journey. And it's interesting to see where you all started to where you are now, and just the different steps and the different life and world events to lead you here. And as you mentioned, it kind of pulled you in rather than you sought it out. And it seems to be a great fit. I mean, we use you guys quite heavily throughout our day to make sure that we stay ahead of a lot of the things. So I know you had mentioned that there was a nursery that was having issues, and you found some of the problems that they had. So with that, what are some of the common issues and problems that you test for within cannabis plants?
As far as the most common pathogens that you're going to run into, there are three big ones. I call them the three horsemen of the cannabis apocalypse. And those are hop latent viroid, which is on top of mind for most cultivators these days. And then two fungal or fungal-like pathogens, Fusarium and Pythium. And so usually when someone's having an issue, 97 to 99% of the time, it's one of those three. Those three pathogens are difficult to spot. There are not very many, or if any, effective treatments, and they can infect plants early on and be incredibly expensive if you don't catch them early. Obviously, there's things like botrytis and powdery mildew that can come later during veg and flower, but those are a little bit easier to control, environmentally. So I would say from a threat standpoint, if you think of nothing else, there's a lot of noise about all sorts of viruses and other things, and they're not there. But I would say those three, if you pay attention, you're covering the vast majority of your risk from pathogens.
For sure.
Madison, is there anything you want to add on those, especially like you said, the big three of the apocalypse? Is there anything on those three things that you can add to how we are utilizing TUMI to test for those?
Yeah. So I kind of look at it the same way. There's three big ones, and it's the same three you mentioned, just because they're the most common and the most severe and the most difficult to treat. Hop latent viroid is our big one, of course. We still pay large attention to the other ones, but hop latent viroid is one where it can go so undetected so easily that we do a lot of preventative testing for hop latent viroid. For Fusarium and Pythium control, we try to control more of like our social and like biological factors to keep that under control and keep our cultivation nice and clean. We still test for it on an ad hoc basis. If we suspect anything strange and we rule out hop latent viroid first, or it doesn't fit the bill for hop latent viroid, but those are our big three too, for sure. And 9 times out of 10, when you have a problem, it is one of those three.
And a lot of time we test to make sure that we are ahead of it before it actually takes over or anything, or just for like a safety net to make sure, hey, everything's good, it's clean, it's thriving like it's supposed to.
Absolutely. And with our operation, we work a lot with client genetics. And so it's a huge, very important safety net for us and our garden. Any client genetics that come through our door, whether it's for us to put through tissue culture or what have you, we test for all those big three before they ever make it into any other stages in the cultivation operation. So, yeah.
Awesome. So we heard from Tassa and all the great beginnings of TUMI and just where they've gone and what they do. What was it like for Endless and you to introduce TUMIGenomics into Endless's operations? What was the conversation like? Was it something you knew from the beginning that this is what you wanted to use? Like, walk me through that.
Yeah. So when I started with Endless, it was kind of right around the same time that Endless started their relationship with TUMI. We had just purchased the TUMIGlow, which is their on-site testing kit. And I was honestly so excited. Like when I first started working here, that was something that I knew I was going to get to utilize. And I was stoked because the cultivation operation I had previously come from, I've dealt with hop latent viroid on a pretty severe scale. And so, we just went through the gamut of testing, waiting a week, getting the results back, knowing that the condition of the garden might be different now than when we sent the results a week ago. So having the rapid results that TUMIGlow offers, which I'm sure we'll get into, but that's one of the biggest advantages of having TUMIGlow, was something super exciting to me. It allows you to test a plant that you're suspicious of and know its disease status within, about an hour and a half to 2 hours, depending on how long it takes you to collect samples, which was a crazy turnaround time for what I was used to with dealing with regular labs. So I was really excited to work with TUMIGlow and Tumi and just everything that they have to offer. So,
Tassa, is there anything you can add about the TUMIGlow and it being like an on-site, like Madison had mentioned, it's pretty immediate. Like you can have results within an hour and a half, however. Is there anything that you can add to that as well?
I think, Madison's right. One of the biggest advantages, of course, is the speed at which you can get those results. And that's really important when you're trying to make cultivation decisions on the ground. Like, should I clone this plant today or tomorrow? And that's just impossible to do with sending things off for PCR. I think the other really important component of the TUMIGlow is that we designed it to be easy. It wasn't meant to be used by scientists. You don't need a vortex and a centrifuge and a bunch of pipettes and a whole infrastructure. It was meant to be used by the regular cultivator. And it was meant to be used anywhere. You can set it up in an office or a hallway. We've even taken it home and cleaned off the kitchen table, and my 8-year-old helped load the samples. So it's meant to be very user friendly, and it also comes with a software component. So the software component can allow you to kind of organize all of those results. You can organize them by strain, by room, by user. You can export results and share them. And so it all kind of comes behind Tumi's firewall. And so it kind of adds some legitimacy and confidence to your, if you have customers buying clones or things. And there's a dashboard that will allow you to look at pathogen testing over time, by strain, by room, by user. And you can kind of see how things are going. And so it gives growers some organizational and some visibility tools, which can be really helpful when you're trying to suss out a problem and figure out what to do next.
I love that you had mentioned it's very user friendly. It's very for on the ground use, and I like you mentioned your 8-year-olds being able to use it, and it's something that growers can be confident using. That they know they're, like you had mentioned, there's trusted results behind it. So I think Madison may have a question for you that she wants to ask. I know she's probably full of questions today.
Yeah, absolutely.
We've talked a little bit about the TUMIGlow. Sorry, I'm going to go ahead and repeat that question because I just smacked myself in the microphone. So we talked a little bit about the TUMIGlow and a little bit about PCR. I know the TUMIGlow does run a little different of a molecular test. It runs RT lamp rather than PCR. I know that has been, it's just something I want to delve into. How can we trust, or if you could explain how we can trust the results and the accuracy of the results from the TUMIGlow as compared to PCR test results? Just shine a little bit of light on that.
Yes, absolutely. So probably the best way to start in this question is to explain just really briefly, what PCR and RT lamp is and how they're similar and how they're different. So both of those technologies are what we call molecular amplification, which basically means you in the sample tube, you might start with 1 or 2 copies of, say, hop latent viroid. So both technologies will find those copies and then take a small part of its genome or its DNA or RNA and make a copy, and then make a copy of that copy, and then a copy of those copies, and a copy of those copies, on and on and on. And by the end of that, you have like billions of copies of that pathogen. And that's why these types of technologies are so sensitive, because once you have billions of copies, finding it in the tube is like, falling off a log or something. There's just so much of it. PCR uses a specific type of molecular machine that requires temperature cycling. And so you have to get these really expensive, they're called thermocyclers, that move the reaction around in a bunch of different temperatures and read everything with this really fancy laser. And so that can give you, quantitative data, which is great, but it also is pretty expensive, and the prep can be, pretty technical. RT lamp, on the other hand, uses an enzyme that doesn't need thermocycling at all. It uses a molecular machine that can do that, like making a copy of the copies at a single temperature. And so it's kind of doing the same thing, but it requires much less technology. And so that makes it accessible to, anybody, anywhere, that can plug in a heat block or a water bath or however you can heat that tube up. One of the drawbacks of RT lamps, the classic RT lamp, is that the technology used to detect that amplification or that copying is happening is non-specific. So if anything happens in the tube at all, it starts to test positive. And that can be dangerous because if it's some sort of non-specific crap going on in the background, you get a false positive. So what we have done with the technology is completely change that detection to make the detection specific. So it's very similar to PCR, or what we call TACMAN PCR, in the sense that all sorts of random crap can be happening in the background of that reaction, but you're only going to see a positive result if the actual piece of material that you're trying to find is present. And so that cuts down a lot on that false positive problem that a lot of RT lamps have. And it also allows us to increase the amount of time the reaction sits in the incubator, which dramatically improves the sensitivity. So we're able to get that sensitivity, between 4 to 5 copies per microliter, which the PCR is 3.5 copies per microliter. So with these changes, we're able to make that sensitivity pretty much comparable. And that allows people to feel confident that when they see a result, it's not a false positive, and that if the pathogen's there, TUMIGlow will most likely find it.
This question might get a little bit more into the science of it, but is that due to your guys' primer design? Does that have to do with the endogenous cannabis gene control that you guys also search for?
Yes, so both of those actually. That's a great question. The test, because of the way we've changed the detection technology, detects 2 things simultaneously. There's an internal control, which is a part of the cannabis genome and living that is expressed in living tissue. And so in every test, you're going to see a color. And that tells you the test is working, that you have good material, that your sample isn't degraded. And if hop latent virus is present, that will also be detected simultaneously. And so you can see both of those things happening at the same time. And yes, and the primer design has changed. And so what we've done with the primer design is basically make them what we call hot start. And so it allows the reaction to kind of sit in stasis until you put it in that heat block, and that keeps non-specific stuff from happening as well.
Nice, very cool.
Awesome. So you talked about a lot of different changes and different, things that have happened over the years. Is there anything that can maybe be on the precipice of the next few years that maybe you're excited about, or something that you want to see kind of change within TUMI, or a product that TUMI may be developing? Is there anything on the horizon that we can see from you guys?
Yeah, absolutely. So I would say there's 2 main things that we're really excited about. The first one is sort of expanding the testing repertoire for the TUMIGlow, and increasing the number of pathogens you can look for, as well as, I would say the most innovative and exciting part is expanding that software. I think one of the biggest problems with pathogen testing isn't necessarily, do I have this pathogen or not? It's, okay, I have it, now what am I supposed to do? And I think that's an issue with agriculture and a lot of industries. And so we're working on making that software kind of a machine learning or a deep learning type approach, so that it can actually take your data look at it over time with various pathogens and other conditions and help you figure out what that next step might be, whether it's water testing or air testing or changing, the phytosanitary protocols and propagation, for example, stuff like that. Yeah. So, and then the other thing we're excited about is next generation sequencing diagnostics. And so that allows us to sort of move past, I'm testing my plant for these 5 things, to just test my plant and tell me what's wrong with it. And we can look at all of the pathogens simultaneously. I think that's going to be really powerful.
So you had mentioned that people who test with TUMI have the question of, okay, now I've got the results. What do I do now? Is there any other common questions or any other common topics or problems that you are presented with that maybe TUMI can look forward to? Or is there anything that people come to you about that's like, hey, do you guys have information on XYZ?
So I think one of the biggest advantages of working with TUMIGenomics is that we approach each facility in a unique way. And so when somebody comes to us, often it's like, oh my gosh, all of my plants have Pythium, or I have Fusarium and hop latent together. What do I do? And so we sit down with the grower and we talk through all of their process. And we go over critical control points like, how do you clone? What kind of tables do you use? What's your water source? Do you sterilize your water? And we kind of suss out where, things might be spreading or pathogens might be entering or any weak points in, all of their SOPs. And then we tailor whatever, thing we recommend to their specific situation. So it might be, hey, let's test your water to see if that's where Pythium is coming from. Or, a lot of times, let's test your cocoa, because sometimes it's not sterilized, and that's how you're introducing Fusarium into your facility. And so it's very unique and tailored to each grower. And I would say my biggest recommendation is to just sit down and think through your whole process when you have a problem. Like, where could this be coming from, and how is it spreading? And it's super important to get a partner, a scientific partner that can help you work through all of those, that understands the biology of the pathogens and how they spread, to make sure that your stuff your changes are effective.
I know we're getting close to time on this thing, so I just want to open the floor to Madison. Is there anything that you may want to ask Tassa? Is there any kind words that you can talk about how we are using their product, like the TUMIGlow, and how it's maybe just been a key component to our operations?
Yeah, absolutely. You mentioned it already, but the way that you guys are with cultivation operations throughout their entire, testing and disease remediation journey, I've experienced that customer support on such a close level as well. I'm constantly getting emails from TUMI saying like, hey, we've been checking out your test results. How are things going at your farm? Have you been noticing any weird things with the way the TUMIGlow is functioning? Is there anything we can offer to help you? And so they're extremely proactive and super helpful and a great resource in that way. The TUMIGlow has been indispensable. We already talked about how, we take in client genetics and we have a tissue culture lab. And so making sure that those are disease-free before ever entering the tissue culture lab is paramount. So having that opportunity is so important, and very thankful for that. Same with being a clone distributor in our market. We can guarantee for our clients that we have disease-free genetics because we do that preventative testing and routine testing, and we can provide those reports of our disease status of our moms and all of that good stuff. So it's just provided a huge layer of insurance and just sense of ease within our operation, knowing that we actually have full confidence with what's going on. I do have one question for you. I was just curious if we could get a little teaser as to some of the pathogen detection you guys have coming out for the on-site testing with the TUMIGlow. I'm really excited to hear about that.
So, I will say that's wonderful to hear how the TUMIGlow has been integrated into your facility, and that's exactly why we started venting this stuff in the first place. So, that's great. That we want to expand and help even more. And so obviously we're going to integrate the big 3. So we have hop latent. We're working very hard to get Fusarium and Pythium added to the list of tests. And we're also looking at adding Aspergillus as well. So people have the ability to test that on site, before if you're worried about a batch or whatever. And from there, we're going to kind of let the literature guide us. So much is being discovered, so many different pathogens, and I'm sure that eminently there'll be new threats, and it's really important for us to stay on top of that and make sure that the people who work with us are aware of it and that we're providing those tools as quickly as possible to sort of prevent those pathogens from causing problems.
Yeah, it's ever evolving, right?
Yeah, no, absolutely. Yeah, no,
sure.
it's exciting in a way because there's so small amount of literature for a long time, and now it just feels like it's exploding. And so, it's kind of cool to see all the new stuff that's coming out.
This has been such a fun conversation. I know, Tassa, thank you so much for joining us. Madison, thank you for coming in and bringing such great questions. I know you two could talk forever on disease testing and the great products that TUMI has. So I just want to send my thank you to both of you for sitting down and having some great conversations today.
Thanks, Skylar, and thank you, Tassa. It's been great.
Yeah, absolutely. Thank you so much for inviting me. This was a great conversation, and I love to hear about all the success you're having with TUMIGlow.
Yeah.
That's awesome.
Definitely. Thank you so much, Tassa.
If you ever make your way to Oklahoma, please stop by. We would love to give you a tour.
No, I would love to visit. That sounds wonderful. Thank you guys so much.
Yeah,
thank you. Take care.
Next time.
All right, sounds good.